Okay, good morning. Welcome to today's Friday seminar, and it is really my great pleasure to introduce Professor Vivianne Vleeshouwers from Wageningen University. And Vivian did his PhD in Wageningen and working with Peter de Wit on Phytophthora resistance. And since then, she stayed in Wageningen and now is a professor in plant breeding. And over the years, Vivianne has established this remarkable program that focusing on developing durable resistance to potato disease. And her research spans from really exploring a collection of wild Solanum species to identify resistant genes that recognize pathogen-derived molecules, to understanding the molecular mechanisms underlying the plant pathogen co-evolution, and all the way to implementing these genes in the breeding programs. So it's really wonderful to have Vivianne here and talking about her research. Please join me in welcoming Vivianne.
Thank you so much. Okay, thank you, and everybody for this very warm welcome, always. Always amazing pleasure to be in Orange and meet many friends. So I will talk about potatoes, I guess. And potato is very important, as you know. It's also very important in the Netherlands. You can see it's important in our history. It's also very important for the future, the food security. But we have another potato eater. This is Phytophthora infestans, causing late blight. And this eats potato leaves very quickly. So you can, these lesions can expand within a few days. The whole leaf is eaten. And around the lesions, you see these spores. You see, I'm horrible with this thing. Here. So you see the spores, and they get showered over the field. And if the field, if it's humid at night, then the next day the whole field is covered, or it's spreading quickly. And within 2 weeks, the whole field can basically be blighted. But here we see a wild Solanum. This is good news, a wild potato, and this stays very happily flowering, Solanum demissum, till the end of the flowering season, of the growing season. And that is what I started working on. So when I started in plant breeding, I collected accessions, about 1,000 accessions from wild potatoes. There's about 200 species, depending how you taxonomically classify them. But it's a very diverse family of plants. And we tested about 5,000 genotypes, initially in vitro, and then later on detached leaves, also in field trials in 3 years. And that led to a collection, that we maintain also in vitro, of these wild potatoes. And this collection was initially selected based on Phytophthora resistance, and later we also start working on other pathogens. And now we use it, basically this is our core business, I would say. And we maintain them, so we have lots of data on all these individual genotypes. So we have an important potato, and Phytophthora is destroying it, so why are there no resistant cultivars? And I think my favorite answer is here. This is on the Chinese wall, the Great Wall in China. And if you don't know yourself or your enemy, then you run in danger. If you know yourself but not your enemy, you may win, but you can also lose. But if you know the enemies and yourself, you can control all the battles. And this is, I think, a wise lesson from a long time ago. So we are studying plant immunity, and we are not only studying the plant, like the plant breeders only would focus on potatoes, let's say, and treat Phytophthora basically as a chemical that doesn't change. That's not the case. And plant pathologists, they would sometimes see the plant as something, that is not changing, but they don't, the genetic diversity on the other enemy, you could say, is incredible. And we need to know both, I would say. So we have, everybody knows this slide, of course. So we have these effectors that are, inside plant cells and recognized by resistance proteins, or NLRs, and then lead to resistance. And also we have these PAMs, you could say, or MAMs, the patterns on pathogens that can be detected by surface receptors, which are the RLKs or the RLPs. I will talk about two of them, and this is leading then to PRR-mediated immunity. So the PRR-mediated immunity is typically more conserved because the ligands are more conserved, whereas the NLR, we know that they are typically fast evolving and get quickly defeated, but they give a very good resistance. So we started this, this effectoromics by looking basically not only at the pathogen but at the molecule, so by looking at the effector. And we cloned it in a Mycobacterium, so using it, so culture it in the lab and infiltrate in the leaves, and then monitor on the leaves if there's a recognition event or a cell death. We can also amplify this by putting a PVX, so a potato virus X, in front of it. That leads to amplification of the response, and it gets very, very sensitive, and it's also very high throughput. You can do a lot of toothpick inoculations on these leaves here. And a third approach is just by having the effector as, for example, as a peptide, which we can do, for example, if there's like pumps, you can have a small peptide that gives the response. And this you can just directly infiltrate in the leaves. So this is a snapshot of one of the screens, one of my favorite slides. It shows here a lot of Solanum species. They are grouped by families or clades. And in the columns you see Phytophthora effectors. So there's lots of the RXLR effectors here on the left. And this is a toothpick assay, so by PVX agroinfection. And then we can quickly just detect the R genes there. So this is a very famous R gene, RPI-BLB1. That was at that time a broad spectrum resistance R gene that was said to be not defeated. At the moment I did my PhD at least. And we see that it's recognizing this effector, AVR-BLB1. And this is also recognized here on this plan. But if you take other effectors like AVR3A, that's recognized in those Solanum here, in Demissum and also in Stoloniferum, and then if we have another one, AVR2, we see other patterns. So we can really, with this heat plot, we can quickly detect where there's potentially new resistance genes. And this is also working for the apoplastic effectors. So this is, for example, elicitin, INF1, a very conserved small protein from Phytophthora that is in the apoplast, and that is recognized actually by quite a few Solanums also. And I come back to that one later. So this effectoromics was kind of a change in the potato breeding, because until then there were only disease tests with Phytophthora isolates, which is very difficult and very quantitative, and you have to repeat a lot. But with the effectors we could quickly detect new R genes in germplasm. And one example that was for breeding aspects very important was that we can circumvent crossing barriers. For example, the BLB1 that I just showed you is recognizing the aviar BLB1, but also Stoloniferum does that. And this Bilbocastanum, the source of BLB1, that cannot be crossed to tuberosum, so to the cultivated potato. You need bridge crosses. That takes 30 years by a previous professor in the department. So by that time, it's basically too late to have your R gene introgressed. But by using now the Stoloniferum homologue, we can do it in one cross, because this is a tetraploid wild Solanum that also has this homologue. And this is really speeding up a lot of the breeding. So other things, and this is actually quite old, but I think this is maybe the concept that we have been working with since then, and it is still, I think, valid. And there was, I told you there's no resistant cultivar, but there was one cultivar, Sapomira, from Eastern Europe, and that it was said to be durably resistant. And it is, and they call it field resistance, because in the fields, Sapomira was doing quite well. And we thought, what would that be? So what type of resistance there? And we wanted to test our effectors. And first we made a cross. So we crossed the Sapomira with RH. This is a susceptible potato line in the lab. And then we tested the F1 progeny, and so those plants here. So here is Sarpomera, SM, and RH, the susceptible one. And we tested the F1 progeny with Phytophthora isolates. And with this strain, we see everybody is resistant. And with this strain, we see, hey, there's pattern coming. And with other strains, we saw even more patterns. So we saw actually lots of patterns. And we found this intriguing. And then we think, hey, there is very specific resistance here. And then we also tested the effectors. So we generated the RXLR effectors. And this was done here together with Sofian at TSL. It became a big collection, also with Paul Birch in Scotland. And this big collection we tested on all these plants. And then actually we found similar patterns. And this is a big puzzle, and it's hard to see on the screen. But if we just focus on two, for example, we have AVF4 is recognized by Sarpomera, not RH, and it has this pattern in the progeny. AVF3A has a slightly different pattern, but it's definitely different. And if you add up the two, you have this pattern. And this is then exactly matching to that pattern. So basically, by puzzling all the patterns, we could detect actually 4 NLR. So these are 4 R3A and 2 more that must be present in that Sarpomera origin. Then, so we had 4 NLR, but we had another funny thing, because actually we knew that we did a field trial also with Sarpomera, and we saw that the progeny is segregating, and it looked quantitative, and this is typically what you say with field resistance, and that was known about Sarpomera. But then we were doing a test. So here we see the time, and so 3 weeks after the inoculation of the field. And this is the AUDPC, so the area under the curve. We see the RH parent is susceptible, gets quickly defeated, and yeah, just 100% diseased. But Sarpomera is sort of surviving quite well. And then the progeny is in between, so it is kind of coming a bit later. And then if we did a statistical test with if there was effectors co-segregated, we found that the lower half, so the resistant half of this progeny, is actually responding to AVR8. So they are the straight lines, and the ones that do not respond to AVR8 are the dashed lines. So in this case, we could even with an effector detect field resistance. But indeed, we have lots of these R genes, we know that, but we also know they get defeated quickly, and we have to protect them, because as soon as there's one R gene found and used by breeders, of course they put it in the field for their production. But yeah, Phytophthora will mutate, so it will get defeated very quickly. So we have to really start thinking on not only how to get the R genes, because we sort of are getting R genes, but how to protect them. And this is also a very old experiment, actually. We tried a proof of concept to monitor for virulence in the Phytophthora population. And for this, we used this BLB1 gene that I talked before, so because BLB1 has a broad spectrum resistance, so at least we expected to see something there. And BLB1 is recognizing aviar BLB1. So that is a gene family in Phytophthora. It's quite diverse. And here's a small tree of aviar BLB1 homologs. And what we see in this tree is that, well, there's a big group here, we call them the class 1. They are very abundant, and there's lots of them. And then there's 2 other classes. And this group 1 is very well recognized by BLB1. And it all makes sense, because this class 1 is very widespread in Phytophthora isolates. And yeah, that completely matches that the RPI BLB1 resistance is broad spectrum. So then we tested lots of isolates, and we also tested which aviar BLB1 is present in those strains. And we found actually very interesting, because there's 2 isolates here in red. Those are the only 2 strains that we found in that experiment that are virulent and that they really can infect BLB1 potatoes. But what was unique about them is that they do not have class 1 aviar BLB1, whereas all the others do have that class 1. So then we designed the PCR here that was able to detect aviar BLB1 and then also have Phytophthora control, and this was all done in quite high throughput conditions at that time. And so we just tested we could test quite a few isolates very quickly. So this is how it went. So we worked with epidemiologists who had these prediction models when to spray for Phytophthora. And the whole aim was to keep the crop healthy till the end of the growing season. And they would take parameters, of course, of the host, so different potatoes, let's say, different cultivars, but also the weather. So if it's 30 degrees, which is rare in the Netherlands, then the Phytophthora doesn't make much chance, but of course it's not always 30 degrees. And in this experiment, we took also the pathogen in the model. So we had this people going to small field plots that were spread in a field and take samples. So as soon as there was a Phytophthora, and here you see my Stoloniferum, so the wild Bilbocostanum homologue, and some cultivars. And if there were lesions in the leaf, then she would pick lesions and post them to Wageningen. And then, so this is how the field looks. So here's the field trial, let's say. It was in 2 locations, in Lelystad and Valtermont. And around the field trial, there's these small bait plots, so where the samples were taking. And she would drive with a car to just pick all the samples, send to Wageningen, so they would arrive here in the morning, a pile of plates, and we have this quick DNA isolation system. And then, by lunch it would run the PCR, and by tea time you would know whether AVR-BLB1 is present or not. And that would then go into the prediction model, and then we could phone to the field trial and tell them, yeah, you have to spray because virulence has been detected. So this is what was in that year. Here we have the 2 locations, and we have Bintje, the very susceptible potato cultivar, and we have BLB1, the wild, basically the Stoloniferum plant, so the wild plant. Bintje needed at that time 10 sprays or 11. And here is some more basal resistance, so it needs less sprays. And this is all done without the monitoring. But with the monitoring, we see that Bintje still needed 10 sprays, also in the other location. But the BLB1 plants did not need any spray, because every moment that if there was virulence detected, then the conditions were so that there was not conducive climate for Phytophthora. So in the end, in this trial, it did not need any spray. So from that, I think we can really decrease chemical control and protect our genes. Although I have to say, of course, BLB1 is a broad spectrum resistance, and at that time it was very broad. Now it's a bit less. There's virulence emerging. But I think also with stacking, even if you have partial resistance, if you stack and you know which virulence is there, we can get probably much further in controlling late blight. So this is a new project that was just funded by the EU, actually, with several labs. And here we want to promote the stacks of our genes, but also to know which avirulence is in the Phytophthora population, and then indeed develop a phenotype, a virulence typing system, like by sequencing probably, and then enter that in decision support. Okay, so we have lots of R genes, and we know these potatoes are full with R genes. And there are many NLRs in the potato genome, and then the wild Solanum are very diverse, and they even more. So I think we can find many more R genes if we want. If I looked, here I have just a few examples. So this is only 4 of them, but we see, for example, they are very restricted. So they occur in certain species that occur in certain regions, but always very, very local, and this is the case for all of them. And I think, there are relatively new genes maybe, so they haven't been spread through all the Solanum, and there's a tight co-evolution with the AVR gene. This is Toluca Valley in Mexico, where Phytophthora is evolving big time and where it has a very ancient origin. So we also have other receptors. These are the PRR, and they are more widespread, and they occur actually in many of the Solanum. So we can see the ELR, the elistin receptors, and also PERU, the other receptor, they are like in about 30% of the wild Solanum all over the place that we tested present. And PERU is even present in many cultivars. ELR is not. But these PRRs are widespread, and they have been maintained through evolution, so that must have a reason, otherwise they wouldn't be so widespread. So ELR is the elistin receptor. That's the first one we identified. So elistins have features of PAM. So they're small proteins of Phytophthora, and actually they have a function in sterol scavenging. So they can take the sterol from the plant of Phytophthora and needs that. And you can see it binds to an ELR is an RLP, a receptor-like protein, and the elistin goes very nice in that shape. And we found that ELR gives a little bit of resistance. So here we have Desiree. So Desiree control, this is a susceptible potato cultivar. You can see the infection efficiency here is about 90%, so it's almost all the time getting infected, and the lesions grow fast. But the Desiree ELR transformant, here are a few of them, and they have reduced infection efficiency, so there's less lesions, and also the lesions expand at lower rates. So we see this leaf is more resistant than this one, but of course it's not the full resistance that you would get by an NLR. But we were looking at ELR at that time also, and we identified, ELR was widespread in all these clades of Solanum, and we identified just by PCR several homologs of them. And we compared them, and it's very highly conserved. It is under purifying selection, and is like a very stable protein gene. So we tested it for recognition of elicitins. So elicitins is also a big gene family in Phytophthora, also other Phytophthora species. So we see Phytophthora infestans here in red. It has several elicitins present and also expressed. Also other Phytophthora with other hosts have elicitins. And we tested them by agroinfiltration here together for cell death with ELR from Microdontum, so the original ELR. And we see that there's lots of recognition by multiple elicitins from infestans, but also beyond the species, so other species. So this is very, very broad spectrum recognition. And then if we tested all the other Solanum, basically we see the same pattern. So there's no diversity happening here. So all of them recognize the same elicitins. So it's very, very conserved, and it's sort of similar what we expect from typical PAM-triggered immunity in that sense. Then we also started working more recently on another receptor, another PRR, and another peptide, and this is PEP13. PEP13 is very well characterized also. So it is, actually it was a topic of the PhD from Torsten Nürnberger, who we collaborate with on this topic also. And it is it's a small fragment of a cell wall transglutaminase, GP42. And the transglutaminases, basically they are a glue of the cell wall. They make it rigid, or they connect the cell wall. And PEP25 is actually similar to PEP13, but it is just a bit longer stretch. So PEP13 is the minimal unit to induce, to elicit defense. And PEP25 just is a bit longer, and we use PEP25 in our experiments because we find it is more reliable, probably a little bit more stable than PEP13 in the potato plants, but the patterns are in general similar. So PEP13 is present in all Phytophthora species analyzed, also in more copies. And it causes defenses in parsley. That was at that time cell cultures where Torsten used to work with, but it also causing defense in potato. And here we see the, so we cloned the receptor. We had 3 candidates here, A, B, and C. And if we co-infiltrate PEP13 with A, B, and C, we find that this is the receptor. We call it PERU for the PEP13 receptor unit. And it gives a clear cell death and Inf1 here as a control. And then we, in stable transformants also you can see it. So this is the wild type Atlantic cultivar potato. There's no response to PEP13 or PEP25, but in the PERU transformants there is a response to both of them. And here in the rosa assays you can see in blue there's flagellin, so there's always giving a response in all the plants, but there's no response to PEP13 in the wild type or the empty vector control, but there's a clear response in the PERU transformants. And with the ethylene assay, you see a similar picture. So we started looking at PERU and also in all these wild potatoes, and we tested quite a few, I think about 500 plants, by infiltrating PEP13 or PEP25. And this is all the plants that we infiltrated, and we see the response to PERU, although there's more plants infiltrated from Bolivia and Peru, whereas the origin of the potato. But in general, we see there's a really hot spot, seems to be here in the high Andes for Peru. And then we started to, and now I have to go back a little bit, because this is work, again, a long time ago from Torsten on PEP13, because at that time he made an alanine scan, and he made some mutations to see which residue is very important for the transglutaminase activity. And that's where these, and then this tryptophan here is a very important one that was never active. And we tested all these mutants and also the wild type PERU on these 500 potatoes, and in about 125 of them there was response to PEP25, and that is this list. So this is the wild type here. All respond to the wild type, and here are the mutants, and then we see all kind of patterns here again. So this is the tryptophan mutant, and then the two other ones. So here, this was not recognized by PERU, but we see all these different specificities again, and here we have a group of plants that recognize more. So even they also recognize the tryptophan mutant. And this is in a Leptophius plant, LPH. So we took the LPH here and co-infiltrated. So this is the original PERU with all the PEP25s, and you see no response to the tryptophan mutant. But here with the LPH PERU, we see a response. So this was very interesting because this is a PRR that is apparently diversifying. And then we went here, we came here to Amir Ali and Sofian, and we did like an amazing study of phylogenomics and take all the RLKs of solanaceous plants. And well, it's a big analysis, but in the end there was this beautiful graph here. You see lots of solanaceous plants. These are not potatoes, here's the tomatoes and up to cultivated potatoes and all the wild species here. And we see actually in gray, you see if there's a PERU homologue. So there's PERU homologues in these. This is eggplant, I think. And then we see PERU here in the Mexican diploids, which is like Bulbocastanum, but all the gray means it doesn't recognize PEP25. The recognition here comes here in the Andes, in the Piurana, the ancient Solanums in high Andes, and then it really exploded in the tuberosa. So we tested if PERU provides resistance, and it does. And again, similar to ELR, it is a quantitative resistance. So here we see the lesion sizes of the empty vector control and the wild type Atlantic plant, and here 4 PERU transformants, and you see the lesion sizes are smaller. And if we do a CRISPR knockout, we see the opposite. So this is the DM, the donor of the first PERU, as this lesion size, but the CRISPR mutant are more susceptible. So it is resistant, but well, when I talk to breeders, they actually don't like it so much. They say, oh, but this is not going to work in the field. And I think maybe we should, study a little bit more. And we hypothesized that maybe by knowing the co-evolution and also some insights in the structure, maybe we can tweak PERU and make it better. So for that, we would need the genetic diversity on PERU and also on the PEP25, which we have. We can do structural predictions, and we can also do very quick transient assays. So this should be a possible way to go. So first we look where there's the positive selection, and this is somewhere, well, there are 11 residues on the positive selection in the ectodomain, and 10 of them are in the concave site. And here is a region based on the domain swapping that we did earlier that seems to be important. And we are focusing a little bit on these 391, 392 positions here. But first, we also looked at genetic variation on the Phytophthora side. And we found this is like 11 natural variants of Phytophthora, of PEP13, that is also expressed during infection. And we tested quite some plants from the tuberosa, but also Piorana, so the old ones, and then the intermediate clade. And we find, actually, so here's the tryptophan mutant is always conserved, and it makes sense because this was important for the transglutaminase activity. But we see some nice patterns here with this arginine. So if the PEP25 has an arginine, we see that there's more recognition. And there's a lot of puzzling also to do here, but we think it's very interesting. And what is especially interesting, that it makes sense in terms of the evolution of these Solanum, because they were born in Mexico. They go, after a while, they go to the high Andes. They like it there, they diversify big time. And there's the first Peru. And then there were some migrations back and polyploidizations going back to Mexico, which explains this small Mexican detection of PEP13 also. But basically, the center of diversity is like here in the high Andes. And well, this is what we are doing at the moment together with Andy and Amirali, everybody in TSL, and the PhD student in Wageningen, Barbara. And yeah, we are looking now on this site where the diversifying selection is significant. And we find indeed here there's a sandwich between the co-receptor and again this important tryptophan. And we see also this arginine is in a very interesting region. And this is all in progress. But we have, a few more things. So Phytophthora, I told you that's my first love, let's say. So we did effectoromics with Phytophthora, but later we also picked up other pathogens, several of them. And one, this is a short moment on, so I have a few words on Phytoplasma and also a few on Alternaria. Because Phytoplasma, I think, has another advantage. Effectoromics has another advantage to study Phytoplasma. Because at potato, there are some emerging phloem limited diseases. So one is Phytoplasma, the other is Liberibacter. And these are transferred by insect vectors. So here you see the Liberibacter that quickly went from America, Hopsake, to, in a few years' time, to New Zealand, Australia, and it's supposed to be Northern Europe. And Phytoplasma is here in Europe. These are not culturable, so that's not very easy. And there's no resistance, of course. But what is very difficult is that there's quarantine regulation. So how are we going to do disease tests if we cannot basically work with it, or at least only a very small unit? So that is why we use effectors here. And now we are doing this with a three-step approach. So at this moment, we target the bacteria directly. So we are using effectors of the bacteria, whereas in the US, they're mainly looking at defense against the vector. But yeah, I think it's also good to look at the bacteria. We can do the insect also later. But we test effectoromics, so we express the effectors, we test them in the Solanum, so the same trick, and if there's a responding plant, then basically we do a grafting experiment. So we transfer the bacteria through grafting. So this is our maintain, so this is tomato having the Phytoplasma. Then we graft it on potatoes, and then you see, sometimes symptoms coming. And then later, this is next year, I guess, we will also complement it with the real insect test. And we get very nice symptoms like the aerial tubers, and here there's the purple shoots. So the grafting is really working well. And I hope to tell you about that next time. And then a few words on Alternaria, which in this case is, you're not doing effectoromics, but Alternaria is a necrotroph. And it is severe on an older potato, on stress plants. It comes late in the season. And traditionally it's from warmer zones, like in the US and southern spaces, but yeah, climate change, so it's getting worse. And also isolates become resistant to chemicals. And of course, there are no resistant cultivars. So here we use the collection again for a screen, a disease screen here. These are like 500 wild Solanum species. Most of them are susceptible. There's quantitative levels. But we have a few here that are very resistant. And one of them is here a Commersoni. And in the close-up, you can see very, very, very strong defense response. It even looked like HR, and that was what we thought when we tried to find, what is the resistance. So we made a cross with another Commersoni and a susceptible Commersoni, and we found a one-to-one segregation in the progeny, very clear. So all HR or all lesions. So we thought, is that the receptor? But no. After fine mapping and everything, we found that those are two glycosyl transferases, and we call them glycosyl transferase 1 and 2 from Solanum commersoni. And actually we thought, how is this working? And then we did a very simple experiment, and I like it a lot. This is just a leaf extract from the resistant and the susceptible commersoni. And you see on the resistant, so added to PDA agar, on this one, the Alternaria cannot grow at all. And on this one, you see a massive spread. And what you also see is that there's even other fungi starting to grow on this one. So we measure the expansion of these lesions or of the colony actually of the fungi in a more clean experiment, of course. But then you can see in the plants with the resistant extract, you see the lesions grow of the colony grow is much less, and that's the same also for botrytis and the same for Fusarium. So this is a very broad spectrum thing. And then we found by purifications and all that this is basically based on steroidal glycoalkaloids, so we call them SGAs. And these compounds occur in potato already, so like solanine and dimecine. And we measured the SGAs from potato cultivar, but also from the susceptible commersoni and the resistant commersoni. And what we see is that the susceptible plants, these two, have triose SGAs, so they have 3 sugars attached to the A glycan, whereas the resistant one actually has 4. It has a more blue color and different compounds. Then we made the transformation, so we put the R1 and the R2 into the susceptible background, and then we see a shift. So these are 3 individual transformants from this plant, and then you see that, well, it becomes a different compound. So there's this SGT1. GT1 is a glucose transferase, so it adds a sugar to the group, and this one adds a xylose. And by adding that sugar, the resistance is conferred. And we see that it works very well against Alternaria. So we see here the wild type, and then you see the transformants. Then, yeah, the lesions get much smaller. And it works also quite broad spectrum to other fungi and even against insects. And here's against the potato Colorado beetle, also important, by looking at the larvae and also the weight of the larvae. So this is very, very broad spectrum. And I think looking at the SGAs that occur in Solanum, and not only potato, but all these wild species, that is really amazing. Because there's a lot of variation in the A glycan and also on the sugar. So you have maximum possibility of specificity also. And what is so funny, that potato and also tomato, they have all these different SGAs, but they're susceptible to Alternaria. But if you make the combination between the chromosome, it has the solanine, so the A glycan from the potato, but the sugars from the tomato, and then you get a resistant plant. So I think we have possibilities here. The trick is they are very toxic also maybe to other insects and to humans. So there's usually braiding against the SGAs. But they can be detoxified, so tomato can detoxify the tomatin during fruit ripening, so your red tomatoes are not toxic anymore. And also this is the case in nigrum, these blackberries, they seem to be edible. And more interesting even, pathogens have evolved different ways to detoxify this tomatin. So here there's lots of pathogens, and what they do is they sort of cut away the sugar. So you go from the tetrose again to a triose, and it's not toxic anymore. So yeah, I think this is a co-evolution also, but at a different level, and it is also highly interesting. So we want to continue this also because I believe there's a very strong potential here for disease control and also pest because of the diversity. Like the receptors, there's also diversity here, and it is kind of a natural pesticide. But because the broad spectrum effect, even fungi, insects, and more, yeah, we have a risk for food security. So normally we want to reduce them, but I think we have here opportunities for metabolic engineering, and we should look, yeah, maybe design a better one or something. And then I think this is the final slide. So to conclude all over, I think the wild potatoes, they are a rich resource for disease resistance. And during co-evolution, or yeah, they have accumulated diverse defenses. So we know they have a lot of receptors, and we use the link between MPMI research and evolution to understand the immunity, but also to maybe make it stronger. So we have lots of NLR. There's so many in the genome, and there's so many more in all these wild species. So they're very abundant and diverse, but we also find they're very clade specific, and yeah, they don't live very long. So yeah, maybe we can work on that. And the PRR are more broad spectrum and give a quantitative resistance, which is not strong enough for a pathogen like Phytophthora under agricultural conditions. So I think now there's a lot happening in the receptor-mediated immunity and the structural biology. So I think there's opportunities perhaps to tweak the receptors or the immunity. And in addition, we also have these metabolites such as the SGAs that work against Alternaria and many other pathogens. It's very broad spectrum, but we here have other opportunities, I think, for metabolic engineering of resistance. And then I should thank a lot of people. I don't think everybody fits here, but yeah, we have lots of nice collaborations, of course. So most of the work of Peru was mainly done by Yeri, who is from Peru, and now in the US. And Lin, who was here, he started that work, and Barbara Schrammel just started her PhD, and she's continuing the Peru work. Jaap is working on the Alternaria, and Jasmin is on the Phytoplasma and the Liberibacter. Dorette is technician doing everything. And then we have nice collaboration, phytopathology, and also groups in biointeractions and bioscience in Wageningen, mainly on the virulence monitoring. And then, of course, a big super interaction with TSL, many people, Sofian and Amirali and Andy, special. And then Thorsten and Nürnberger in Tübingen with Lisha, they contribute a lot to the protein work at that time with Peru, and then many other collaborators. And I thank you for your attention.
Thank you very much, Vivianne, for a great talk. So we invite questions from the audience, and students and postdocs first, please.
Hello, interesting talk. I just wanted to point out, like, what made you discern between differences in Mexico specifically, between you specified that they were local, but as I see from the points in the different mapping of Mexico, it's actually not that local, it's more widespread. I think it's just a mere part of like map scale, because I saw, well, I'm from Mexico and I recognize the states, and some states are like 100 kilometers away. And and I suppose I think it's more local, but it's not between the distance, but it's more because in Mexico, different from Peru, Argentina, and other Latin American countries, is that we are in a zone that it's a country with a lot of microclimates. So if you just travel 30 kilometers away, you're in a forest. You travel 60 kilometers and you're in seaside. So I just wanted to know if this type of microclimates is inferring some epigenetic changes in the plant of potato, rather than being something genetic. And because obviously potato can grow in different type of climates, depends on the soil, of course, but as the plant grows, also is changing because it has to endure different type of climates, different type of rain seasons, and in Mexico we have those. So it's just a mere question how much epigenetic changes are involved in this type of resistant genes in potato. Thank you.
Okay. Yeah, I think, well, to be honest, I haven't studied epigenetics, but what I meant with local, actually, maybe local is not the best word then, but it is, if you compare to the PRRs, they occur all over the region where Solanum species grows, so from the very South America to high north of Mexico even, but that also counts for the phylogenetic groups. So if you look at the more ancient Solanum from Mexico, the diploids, the Bulbocastanum, and even the older ones, up to tuberosa that are, so higher in the phylogenetic tree. So local, I meant, restricted maybe. So not local climates, because indeed, they are very different.
And how did you assess the phylogeny if, for example, there's historic times that Aztecs from Mexico used to commercial products from Peru, from Quechuans, and has there something to account that type of early agro communication between those type of cultures in the past?
Yeah, could be. I think this, I think this evolution of these solanums is very old, and I think what is actually making it restricted or local to certain, restricted to certain clades, I would say, is that it occurs, for example, in a certain species. So maybe they're younger in the evolution. They're not like if here's your tree and here's the potato, but the old one. So if like Peru is already, no, ELR is very old. It came a long time ago and it fixed and then it stayed. And Peru came later, I think, and it started to, it's still diversifying, but the origins are much later in evolution, before the speciation of all these clades. That's why I think that we see the NLR only in a very few plants have, for example, BB1 or R2. Only like R2 is very fixed in Mexico, or fixed, it's in Mexican Solanum only. There's another NLR that also recognizes AVR2 that occurs in Peru. It's Mojiquense, but it's very different. It's different NLR. So like no relation at all. So AVR2 is recognized in two regions, but the NLRs
they are quite different.
Are just younger, I think, in evolution. Yeah.
Thank you.
Yeah, there's a question from there, Sam.
Great talk. Thank you for that. Have you got any theories why there's no resistance to phytoplasmas? And I think you said there's also no resistance to the Liberibacter.
Yeah, I don't know how far we have looked, right? I mean, and also, so we just started, and I have to say with Liberibacter, but this is very preliminary, we have very nice patterns of recognition. We found like responses that look promising, but we still need to do the grafting there. We still don't have that bacteria, so it is too early to say we find something. But I think that is promising. With the Phytoplasma, we started a bit later, and we tested some effectors, we do see some responses, but at this moment, it's too early to say whether we find a receptor. Also, we don't know if we would find a receptor, right? We don't know if receptors control these pathogens also. But so far, except Alternaria, and also for Alternaria, they're NLRs. So I think so far these Solanum are so full with these receptors, I want to give it a try, right? And then, but I think in parallel, there should be other ways to control Phytoplasma. Well, we have to find it, right? Yeah.
Vivianne's question there says.
Hi, thank you.
Hi.
Great talk. I was interested in your thoughts on the PRR-mediated immunity, and what do you think about maybe the durability of resistance? Should you be stacking PRRs or even stacking them with NLRs?
Yeah, I think, well, actually we tried to stack ELRs. We stacked PERU with RP23, and we found there is a slight enhancement of resistance. So we find, especially if you test with the effectors, so with PEP13 or with the NLP20, then you see that the ROS curves, really, I mean, they have their own ROS curve, and in the double transformant, you see the pattern is like complementary, so it is additive. But the resistance is only slightly enhanced. And we tested also with several isolates, then the PERU resistance is consistent, it always gives a certain degree, but the RLP was not very stable in these experiments. So we also do tests with ELR and RLP23, and also we find a slight enhancement of resistance, but it's never as high as an argine. And I think maybe it looks like the pathways get saturated with these species, with these PRRs. That's what we were discussing with Torsten. Maybe it's just, this is it. We don't know. I think we should, combining with NLRs, but then with the right one. So there's a lot of interactions that we don't know of. And I think indeed we should know which one to combine with what. But still, I also think maybe effector suppression is interesting. Because, we see the there's many RXLRs that suppress PERU, and also we have RXLRs from Phytophthora that suppress PERU. We have RXLRs that suppress ELR. So there's a lot going on, and I think the complexity, we really should look at all the specific interactions we can get there, I think. But also, I would say, not just breeding, also the IPM. I mean, in the field, and have to predict what the pathogen is doing, and really take the right receptors out there and not burn them down one by one, because then we lose them all. Yeah.
Thank you, Cess. There's questions there?
Thank you. Fascinating combination of the co-evolution of pathogen and host. Going back to these combinations, do you have the ultimate target of the effector is not the NLR, and it will be in a certain compartment. It will either be apoplastic or it will be cytoplasmic. It will be targeting the chloroplast or the mitochondrion. Are you able to cluster these and then group your NLRs according to the targets, following sort of HIV kind of chemotherapy, that you can target different routes? Because it doesn't matter if you knock down an effector that has been detected in one clade, but if you're stopping it attacking all the key things, how far have you got with classifying where the effectors are targeting?
Yeah, that is not known from all the effectors. Yeah, that is happening. I think several people work on that. From some effectors we know the target. Actually, there are many targets. Avirulence is like a hub of, it seems to target everything. So we should do that indeed. It's kind of, I would say, pyramiding origins or pyramiding receptors, but then in a very educated way. Yeah, ultimately we want to know that all and do it very precise, but it's not that advanced at the moment, I think.
If I may, thinking about your wily pathogen, for your peptides, following the sort of chitin binding elicitor that you get with some fungi, do you see in any of your pathogens proteases or peptide binding that is stealthing this signal for your PIRs?
Yeah, we haven't looked at it so far, but yeah, could be. I think there are some proteases involved in, some bacteria I know of that use it, but we haven't looked in that so far. But could be.
Thank you. Fascinating.
Yeah.
Saskia there. Saskia.
Thanks for this wonderful talk. I had a question because you talked a lot about how Phytophthora co-evolved with the potato, and maybe for at least Phytophthora infestans, and also Phytophthora infestans is a specialized pathogen, right? And then also the Liberibacter, the zebra chip one at least, may also be very specific to potato and tomato, right? So then in that case you have found resistance genes, maybe also for the Liberibacter, but I wonder how that works for more general pathogens, for pathogens that may have not co-evolved so much with potato. I don't know if Alternaria falls in that category, but and whether then, how then the NLR screening works, so how you would that approach it? With the potato set.
For Alternaria, indeed, we didn't expect to find it because it's a necrotroph, although there are analyses against another Alternaria, so we thought, well, you never know. But we found this glycoalkaloids and we thought, this is so interesting, so we went there. And for the Phytoplasma, actually, we don't know. So at this moment, because it's very easy for us to screen these effectors, but I think for Phytoplasma we can have other possibilities also, and we should look at that. And I think if we can now do the disease test also, and when with the grafting it starts to work, although it's a lot very time consuming. So maybe we find other mechanisms. I'm very open to any mechanism. But for us, it's very easy to start with the effectors. That's why we did it, and we just started, so we'll see.
More questions? Yeah, Sophie here.
Vivianne, I'm curious about whether you have anecdotal evidence that some of the weaker resistance, like the PRRs and maybe some of the weak NLRs, that when you make transgenic plants and potentially overexpress them, you get higher level of resistance compared to the original introgression or the original source of the resistance. The reason I'm asking is, with the weaker resistance, especially the PRRs, maybe overexpressing them could lead to higher resistance, and that might be opportunities there for promoter engineering and things like that. Any anecdotal evidence for that?
No, so far, because the Peru also we have with 35S, and that's quite high. I don't think you can go much higher. The NLRs normally we do with the endogenous promoter, so they're not that much overexpressed. I don't know. I don't have evidence for that.
But is the transgenic 35S Peru has higher level of resistance compared to the original Peru accession?
Oh, but that is hard to compare because there's lots of stuff there. Although it is, it is the yam plant, right? So that is a susceptible plant. And if you knock out Peru, it becomes more susceptible. This is also funny because Peru is in most cultivars, 90%. So breeders say, well, this is not useful, but imagine if there wasn't Peru, right? But I think also there you could engineer like promoter or tissue specificity. These things, there's lots of promoters and things we could play with. Maybe it's a way also,
So maybe I wanted to ask you a question, Vivianne. Now you have identified all these wonderful resistance genes, and where are we on breeding resistant potatoes? And what's the best way of putting this really in the production? And now, we don't have to worry too much about all these disease. What's the current practice?
Yeah, well, these days, so this point we got. I cloned quite some NLR, and then the GM thing is not so easy in Europe. So that was a bit disappointing. What was very nice is that there was also hybrid breeding coming. So that is moving faster. And now we see all the genes are getting introgressed. So I think that's a faster way. But also the gene editing, I think it should come. Because without it, it's not going to work in Europe. We need potatoes, yeah.
Stacking, so we talked about the stacking.
Yeah.
So are there real, I guess, programs to stack the PRRs and NLRs, or selected PRRs and selected NLRs, and maybe some of the toxin production genes, and to put all these together and have the IPM?
But if we want that, we need GM or gene editing or the novel. Yeah, it's going better. I think Europe is hopefully gonna accept. Because without that, just with breeding with potatoes, that's impossible. They're too diverse. Yeah.
Thank you, interest of time, and let's thank Vivianne again. It's a wonderful talk.